Good morning, welcome Sham Stars. In continuation of chapter 9, we have already done what are enzymes and properties of enzymes in previous video. Today we will learn about factors affecting enzyme activity and classification of enzymes in this presentation. In biochemical reactions, activity of enzyme is affected by many factors. These factors are temperature, pH, substrate concentration, and inhibitors. In coming slides, we are going to learn effects of these factors one by one. Temperature is a very important factor to control enzyme activity. Enzymes are most active at an optimum temperature. Usually it is 37 degrees Celsius in humans. Enzymes show little activity at low temperature, and they lose activity at temperatures above 50 degrees Celsius. We know enzymes are protein in chemical nature, so at high temperature, the nature of protein is changed. Or we can say at high temperature, enzymes are denatured and they lose their catalytic property. Enzymes have an optimal pH. However, the optimum is not the same for each enzyme, but enzyme shows maximum activity at optimal pH. Activity declines both below and above the optimum value. This slide shows enzyme and substrate concentration. With the increase in substrate concentration, the velocity of the enzymatic reaction rises at first. The reaction ultimately reaches a maximum velocity, which is not exceeded by any further rise in concentration of the substrate. This is because the enzyme molecules are fewer than the substrate molecules. And after saturation of these molecules, there are no more free molecules of enzymes to bind with the additional substrate molecules. Here in this graph, we can see by the green cross point of saturation. This is the point which is at the maximum speed. Further, substrate concentration has no effect on the enzyme activity. Other than temperature, pH, and substrate concentration, the activity of an enzyme is also sensitive to the presence of some specific chemicals that bind to the enzyme. When the binding of the chemical shuts off enzyme activity, the process is called inhibition and the chemical is called an inhibitor. Inhibitors are of two types, competitive inhibitors and non-competitive inhibitors. Competitive inhibitors bind the active site of the enzyme competing with the substrate, whereas non-competitive inhibitors bind the another part of an enzyme, causing the enzyme to change shape and making the active site less effective. Let's try to understand inhibition with the help of this diagram, which is shown in this slide. The above one is of competitive inhibition, where the shape of the inhibitor is as same as of the substrate. So there is a competition between the substrate and the inhibitor molecule to occupy the active site of the enzyme. When inhibitor molecule occupies the active site of the enzyme, there is no reaction. In non-competitive inhibition, the molecule joins the enzyme in the other site. It is not the active site of the enzyme molecule, but the other side of the enzyme, and it changes the shape of the active site. Again, there is no reaction as a result. Thousands of enzymes have been discovered, isolated and studied. Most of these enzymes have been classified into different groups based on the type of reactions they catalyze. Enzymes are divided into 6 classes. These classes are Oxidative Reductases, transferases, hydrolyzes, lysases, isomerases, and ligases. The first class is Oxidative Reductases. These enzymes catalyze oxidoreduction between two substrates, that is removal of hydrogen and addition of oxygen to hydrogen forming water. So in this reaction, one substrate is reduced and other substrate is oxidized. The second class of enzymes is transferases. They transfer a group of atoms from one molecule to another. For example, transaminases transfer amino group from one molecule to another. Third class of enzymes is hydrolyzes. These enzymes catalyzing hydrolysis of ester, ether, peptide and glycosidic bonds. The fourth class of enzymes is ligases. Enzymes that catalyze removal of groups from substrates by mechanism other than hydrolysis, leaving double bonds. They catalyze the cleavage of carbon-carbon, carbon-oxygen, carbon-sulfur and carbon-nitrogen bonds by means other than hydrolysis or oxidation. The fifth class of enzymes, that is isomerases. It includes all enzymes catalyzing interconversion of optical, geometric or positional isomers. The last class of enzymes, that is ligases. Enzymes catalyzing the linking together of two compounds, for example, enzymes which catalyze joining of carbon-oxygen, carbon-sulfur, carbon-nitrogen and phosphorus-oxygen bonds between. DNA ligase is involved in DNA synthesis. Let's talk about last topic of chapter biomolecules, that is cofactors. Enzymes are composed of one or several polypeptide chains, that is proteins. The non-protein part of the enzymes is called cofactor, which make the enzyme catalytically active. The protein portion of the enzyme is called the apoenzyme. Cofactors can be divided in three categories, that is prosthetic groups, coenzymes and metal ions. Prosthetic groups are organic compounds and are distinguished from other cofactors in that they are tightly bound to the apoenzymes. For example, in peroxidase and catalase, which catalyze the breakdown of hydrogen peroxide to water and oxygen, heme is the prosthetic group and it is the part of active site of the enzyme. The next category of cofactors, that is coenzymes. Coenzyme is required by some enzymes, an organic molecule bound to the enzyme by weak interactions or hydrogen bonds. Most coenzymes carry electrons or small groups. Many have modified vitamins in their structure. The third category of cofactors is metal ions. A number of enzymes require metal ions for their activity, which form coordination bonds with side chains at the active site and at the same time form one or more coordination bonds with the substrate. For example, zinc is a cofactor for the proteolytic enzyme carboxypeptidase. So children, this is all about the enzymes classification, factors affecting the enzyme activity and cofactors. I hope you must have understood. Thank you, Shem Stars. That's all for today. You can drop your queries on Google Classroom. Take care.